Hey fellas, and welcome to lesson 8 of unit 4 topic 1, and we're looking at gene regulation in this PowerPoint. Okay, now there's quite a number of syllabus dot points that need to be met in this PowerPoint, and you'll notice that a lot of the words in pink here are more of the lower level cognitive verbs. So they're not asking you to analyze and interpret or explain. It's just asking you to recognize, identify, recall. So my sort of take from that is that you need to know it, but you may not need to know all of the ins and outs and the complexities. It is a very complex topic that you could go right into detail with. I'll just go over it quickly because we'll talk about this again at the end. So you need to be able to recognize that the purpose of gene expression is to produce functional gene products, and that could be protein or an RNA, and we've talked about that already. You need to know that that process can be regulated and that it's used by all known life. The second point is that you need to identify that there are factors that regulate the expression of genes. So genes aren't all just on at the same time and doing their thing. There are factors that are going to regulate what's on and what's not. And we'll talk about that in different contexts. You need to recognize that differential gene expression, controlled by transcription factors, regulates the differentiation of cells. So this is looking at how we form tissues and produce in development, and that's really highly regulated as we're developing. And the last one, you need to recall an example of a transcription factor gene that regulates morphology. So we're going to look at the HOX genes, and we're going to look at the sex-determining region Y genes, which allow us to differentiate into males and females. All right, so let's go into regulation of expression, section 5.3 of Pearson Biology. So hopefully by now you're well aware that gene expression refers to transcription and translation. This process is heavily controlled because not all genes are on and working and making proteins or RNA units at a given time. In certain points in life, certain genes are switched off because we're not using them anymore, or we don't need a particular protein to be expressed. So this is hugely regulated, and this is the point of this PowerPoint, is looking at the regulation of our transcription and translation. We've talked about the human genome and how we have a huge number of DNA that controls all of our genes and traits. The cell can express a selection of those genes at a given time, and that's going to determine the proteins that our cells are producing. But it doesn't do that for every gene all on and transcribing and translating all at the same time. So gene expression is really tightly regulated, and there's multiple mechanisms that allow that to happen. Comparing prokaryotic and eukaryotic cells here, the process is much more complex in eukaryotic cells, just purely due to the complex nature and how much DNA we have in eukaryotic cells. Prokaryotes, remember, their DNA just floats around inside the cell, and it's in that circular plasmid DNA, much simpler. You may remember us talking about transcription factors when we were introducing transcription. These are really important in the regulation of gene expression. Transcription factors are proteins that are going to control whether a gene is expressed or not. And they do this by either binding to a region close to the promoter, or they can bind specifically to RNA polymerase and alter its activity. So they can either induce gene expression, so activating it, or they can repress it. They can actually stop the gene from being able to be transcribed. So they're kind of like these little molecular switches that can turn specific genes on or off, depending on what the body is needing. This is an example from Khan Academy here, and you can see this example here. Here's a strand of DNA, and this particular transcription factor is going to act as an activator here. Now see, our RNA polymerase has binded. This activator is going to attach itself to the RNA polymerase, and it's going to assist it in beginning transcription. So that's going to allow it to go ahead and proceed with transcription. This one here is an example of a repressor. And so what happens is the repressor can bind to a particular region at the start of the gene, and it can block the RNA polymerase from even being able to bind and begin transcription. So it kind of acts like an enzyme inhibitor where it binds and blocks the activation. So basically switching off that gene. This is an example out of your textbook of a repressor protein, and they're quite complex proteins, and it's the pink structure here that's bound to the DNA. You can see the DNA double helix in the background, and the repressor is binded to it, and it's physically blocking access to the DNA. So that whole initiation part of transcription where the RNA polymerase goes and binds can't happen. So that gene is switched off, and it's not going to be making the product from that gene. Now, in terms of genes, we have two main types of genes, structural and regulatory. Another term that's used for genes is constitutive genes. A constitutive gene is a gene that's always switched on and it's expressed continually. Not many of our genes are constitutive genes because they're often either switched on or off depending on what's going on in the body. Most genes need to be induced or repressed, so switched on and off, using our transcription factors that we mentioned. So in terms of our genes, the structural genes are genes that are going to code for proteins and RNA that are not involved in the regulation of genes. So this is the bulk of what we talk about when we talk about translation. We're making useful proteins, that could be enzymes, proteins that are in charge of making particular pigments in our skin, could be so many different things in our body. And also our RNAs, so our tRNAs and so forth. So they're structural because they're involved in actually making structures, but our regulatory genes are genes that are going to be coding for our transcription factors. Now, don't forget, transcription factors are types of proteins, but the genes that are going to code for our transcription factors are called regulatory genes because they play a particular role there. Over to the right here, you can see a little diagram where you've got a regulatory gene here, and it gets transcribed into mRNA and translated into a transcription factor. And then those transcription factors can go off and assist in the production of a structural gene because they're going to be assisting in the switching on or off of those genes. Now, one really common model, which is described in your textbook, to look at how gene regulation works in an example is the lac operon. This is a simple model to use because it's looking at a prokaryotic model, and it's looking at an operon, which is a section of DNA that is under the control of a single promoter, but that DNA codes for several different proteins. Now, this lac operon is found in E. coli and other bacteria, and it is an inducible operon. So that means that we can switch it on or off to produce those genes, to produce those proteins, and we can do that using the transcription factors that we've talked about. Now, this operon works by the presence of lactose, which is a sugar that is used by E. coli. And when lactose is present, it's going to allow that operon to be active, and it's going to express 3 different structural genes. And those structural genes code for 3 enzymes, which are there to break the lactose down into smaller sugars. So, for example, if this lactose sugar is present in the E. coli, that means that this lac operon is going to be active and switched on, and it's going to allow the lactose to be broken down into glucose and galactose, which are smaller sugars. Now, there's no point having this gene switched on if there's no lactose available, because it's wasting energy and it's going to be transcribing and translating into these proteins, enzymes, that have no lactose to act on. So this is why genes switch on and off at particular times based on the environment and what's going on at a cellular level. So this is what the lac operon looks like. So as I said, it's got a promoter region, which has its one singular promoter for those 3 different structural genes. And remember, that's where our RNA polymerase is going to bind to begin transcription. It also has an operator, which we call the place where a transcription factor is going to bind. So in this case, the transcription factor is going to be a repressor. So this is going to bind when we don't want the gene switched on. Then we have 3 different structural genes, and they're those genes I talked about that are going to assist in the breakdown of the lactose, and they're called LAC-Z, LAC-Y, and LAC-A. So 3 different enzymes that are going to help with the breakdown of lactose. That entire unit is what makes up the LAC operon. So one promoter with multiple genes regulated by a transcription factor. Okay, so next to the LAC operon, and it's not shown in this diagram, there's a regulatory gene which is going to code for that repressor transcription factor. That gene is expressed continually, so that means that the repressor protein is always present. Now, in this case, in this particular example of the LAC operon, because the repressor is always present, it's going to be bound to this operator, and it's going to block RNA polymerase from binding, and therefore we're not going to get these structural genes being produced. And so that's the normal way that this gene looks when there's no lactose present in the cell. That transcription factor is binded, RNA polymerase can't go across, and the structural genes aren't made. Now, how this works, and I've kind of blocked it with my big old face here, is that when lactose is present, lactose itself binds to the repressor, and it stops the repressor from being able to bind and stop the action of the RNA polymerase. So therefore, the RNA polymerase can bind as normal, transcribe the DNA, and then move on to translation where the structural genes are produced. So it's actually that when lactose is present, it stops the repressor from binding. When lactose isn't present, the repressor is bound. So this whole operon is regulated by that LAC repressor being present or not. Okay, so here's an example of that visually. So when no lactose is present, transcription is blocked. And that's because we have the repressor protein binded here. And it's going to stop RNA polymerase from binding. And so the gene to make our special enzymes is not going to be forming. So we're not going to get any mRNA. When lactose is present, so that's shown by this yellow molecule, the lactose binds to the repressor and removes it. It stops it from binding to the operator region, and so therefore transcription can occur. We get our mRNA, moves on into translation, and our genes of interest are going to be produced, which are going to break down the lactose. Now, another really important type of gene is a master regulatory gene. Now, we know what a regulatory gene is now, but a master regulatory gene is a gene that is going to be regulating the genes during embryonic development. So it's going to control what's turned on and what's turned off during the development. So that can have some major effects on the anatomical structure of an organism. So how they work is that they turn other regulatory genes on or off, and so that's going to lead to making transcription factors that are going to regulate other genes. So they're hugely complex because they're going to be regulating all the other regulatory genes that are going to control the genes during development. One singular master regulatory gene can control the development of a really complex structure. So, for example, the eye, the nervous system, they can be controlled by one master regulatory gene during development. Now, one particular type of master regulatory gene is called the homeotic genes. And these control the structure and organization of the body segments during development. Now, over to the right, we're going to have a look at this because the common housefly, the Drosophila, is a really common model that's used to look at how these genes work. Now, a normal fly would develop a single set of wings in a particular zone along the body of the fly. However, when the homeotic genes are interfered with, we notice that there's going to be strange things happening. So we've got a duplicate of the wings, and the second set is located a lot lower. So the homeotic genes control those anatomical structures and where limbs and things like that are positioned during development. So a really common example of the homeotic genes are the Hox genes, and these are mentioned in the syllabus as an example for you to know. So the Hox genes are master regulatory genes, and they affect the spatial pattern of expression of genes. So their job is to determine the entire plan of the body from the head to tail axis, and so they determine what structures go where, and they also control which genes are on at specific times of development. So that's going to mean that certain tissues form at certain times of development, and from a zygote to a fully developed fetus, if we're talking humans, these genes are what are controlling the tissue development along those time frames. So these are huge regulatory genes, because if there's mutations in these genes, that can lead to errors in the placement of body parts. Now, this example on the right here is showing the Drosophila example again. And what they do with these is they actually put mutations into these flies to look at the effects of particular genes. So A up the top here shows a normal fly, and B has a mutation in the Hox genes, and that is going to result in this fly that has legs growing where the antenna should be. So see how they've got these legs protruding out instead of their little antenna. So huge structural changes can come about from these Hox genes. So the Hox gene family has been used for years as a model to look at evolutionary patterns. What they've found with these genes by looking across many, many different species is that there's a huge degree of similarity in those genes, regardless of what different species we're looking at. So they often use the Drosophila as a model of this, and on the right here it's comparing the Hox gene in the Drosophila, the fly, and the human. Now you can see that there's different points along the genes where there's going to be genes located, and the color coding corresponds to the part of that body line from head to tail. So see how these genes earlier on, this is going to correspond to the upper neck, lower skull in the human, and it's that real front section there on the fly's head. Moving towards the middle, you've got this part which is probably around the lumbar part of the human spine, or probably a little bit higher, the thoracic sort of region, and that corresponds to a similar region in the fly. So these genes really are highly overlapping if we talk about ancestry and evolution, because of the homology among all the different species with these genes. Okay, now the other example of regulatory genes is the SRY genes. Now this is going to determine the sex, and in humans we know that the female has 2 X chromosomes for chromosome pair 23. The male has an X and a Y. Now how sex works is that the Y chromosome carries this regulatory gene, and it's called the sex determining region Y gene. Now this gene is going to code for transcription factor proteins that are going to have male genes switched on. Okay, so basically what that means is for females, because they lack that Y chromosome and they lack this sex determining region Y gene, they continue to develop as a female. There are no transcription factors present to switch on male genes. For males, it means that they start developing basically as a female, and when this gene is doing its thing, it's going to produce the proteins that are going to cause the male genes to switch on, and the male continues to develop as a male. Some new research has recently come out and suggested that maybe this gene isn't switching on male genes, but maybe it's switching off anti-male genes. So it's not really 100% well understood how that works yet. Recent studies in mouse models has found that if that gene is not expressed at the correct time, sex differentiation doesn't occur. So there is some linkage between this particular gene and sex differentiation, properly separating into distinctive male and female. So there's a time frame where that gene needs to be expressed for this to work properly. Now, we've looked at how transcription factors proteins can affect gene transcription. So they're hugely involved in regulation. Epigenetics is looking at other mechanisms that can alter gene expression without interfering with the DNA sequence. So this is another example of things that can affect gene regulation outside of those transcription factors. And how this works is the structure of the chromatin can be altered, and that can be passed on to future generations without having any effect on the actual DNA. So the term epigenetic inheritance is talking about inheriting traits that have been passed on due to changes in the chromatin structure rather than the actual nucleotide sequences. So this is an example of an environmental inheritance rather than an actual genetic change to the DNA. So a common one that is seen is that a methyl or phosphate group is added to the DNA or the histone proteins in the chromatin, and this changes the way that that DNA coils, and it changes the way gene expression works. So normally, for example, if this is what the DNA looks like, you've got your DNA wrapped around the histones to form the chromatin. An example of epigenetic modification would be the binding of a methyl group or a phosphate group, and that is able to be inherited and passed on to the offspring. However, it doesn't actually change the structure of the DNA. It just interferes with how the coiling works within the chromosome and how the genes are expressed. Now, another point when it comes to the environment influencing the gene expression is looking at age and how age affects the traits that we express. And one way that this is done is by studying identical twins. These sorts of studies are really useful because we can look at two people that share the same genome, and if they are raised together and share the same environment, then often any differences in their traits or their phenotypes have to be due to some type of epigenetic modification. Because their genes are the same, we can sort of determine that any differences between them can't be from a genetic sort of alteration. It must be something else that's not genetic, which would come back to our epigenetic changes. So this is something that's looked at by epigeneticists, and it can help to explain why one twin might acquire a genetic disease while the other doesn't, even though they have the same upbringing, the same genes, and much the same sort of in every other variable. Now, often these differences in epigenetics happen more so over time, because as the human ages, they get different characteristics because of the environment and any possible changes that might make to the chromatin structure, which we just spoke about. On the right here, this is a chromosome, looking at chromosomes across 3-year-old twins versus 50-year-old twins. And it's pointing out that the yellow here is where the twins have epigenetic tags in the same place. So they would share very similar traits on those chromosomes. However, over time, with the 50-year-old twins, the red and green is where they have the epigenetic tags in different places. So you can see that they don't line up the same way as they did when they were 3. So this suggests that over time, within the environment, we get these epigenetic differences which exist in the chromatin. Now, there's some videos within this PowerPoint that would be really handy for you to watch. There's some quite lengthy ones, so I recommend having a good look at these in your own time. All right, so now having actually done this lesson and going through all of that important stuff, hopefully you can recognize that gene expression's job is to synthesize a functional gene product, that we can regulate this process, and that all known life uses this process. Hopefully you can identify that there are factors that regulate expression. So during transcription and translation, looking at our transcription factors for the products of other genes. So how we can, genes can produce products that can then, such as transcription factors, that can go and bind and affect transcription and translation. And environmental exposure, so age and changes over time. Hopefully you can recognize that differential gene expression controlled by transcription factors regulates cell differentiation for tissue formation and morphology. So when we were looking at those Hox genes, and examples of these genes that regulate morphology. So looking at the Hox genes and the sex determining region Y. So there's quite a bit of goals there that are covered. So please make sure you have a look and maybe watch some of those videos and delve into this in a little bit more detail. Now, in the syllabus, it also does say, when identifying transcription factors in gene regulation, you do not need to refer to operators, promoters, regulators, enhancers, silencers, insulators, Tata boxes, polyadenylation, and DNA methylation. So any of those terms that I've mentioned, it does say that you're not required to use that terminology. It also says students should recognize gene expression in the context of an example. They are not required to explain or describe the mechanisms of this process. So that was what I was saying a little bit earlier about how those dot points are simpler cognitive verbs, and the syllabus specifically says you're not required to explain or describe mechanisms of gene expression. So I guess interpret that how you will, but that's there as well. Happy studying!